Alright guys, in this short video I'm going to introduce the special cell division type known as meiosis. That's the word we see written right up here. That's how you pronounce that. It's spelled meiosis, but it's pronounced meiosis. So what meiosis is it's a cell division, and so like all cell division, you start with one parent cell and you wind up with two daughter cells. Most cell division that happens during growth and development is what we call somatic cell division, and it involves a cell division type that is shown right here, mitosis. Mitosis is somatic cell division, and that's how we generate all of the trillions of cells that make up our adult body from one zygote created when a sperm and an egg fuse during fertilization. The only time that meiosis occurs is in the gonads when we generate the sex cells. Meiosis, unlike mitosis, is a two-round division process. Like all division processes, we have to replicate the entire genome before we can divide it. And in AMP1, we talked at some length about how DNA replication occurs. In meiosis, we have one round of DNA replication, which means we take our 46 chromosomes and we copy them. In meiosis, we divide that cell 2 times. So what we wind up having is we replicate our chromosomes, we divide them once, and then we divide them again. So after 2 rounds of division, we wind up with daughter cells produced, which are going to be the gametes, that have half the amount of genetic information as the original parent cell. So meiosis, we usually describe it as 1 round of replication, 2 rounds of division. Mitosis, on the other hand, 1 round of replication, 1 round of division. So in mitosis, we produce daughter cells that are genetically identical to the parent. In meiosis, we produce daughter cells that are genetically distinct. They're different, right? So as I mentioned down here, one of the main functions of meiosis is to reduce the number of chromosomes in half. And we call that, for that reason, we call meiosis the reduction division. We go from 2 full sets of chromosomes, which is given by this formula here, 2n, 2 full sets of chromosomes, to 1 set of chromosomes. The 2n, or the diploid arrangement, that's when we have 2 sets, and n is haploid, that's 1 set of chromosomes. And we'll talk about just how exactly this introduces genetic diversity in a little bit. Yeah, we'll get to that in a second. Alright, so for this I'm just going to make a little drawing here to remind us of a few things about chromosome structures. Chromosomes are a hybrid molecule that consist partly of protein and partly of nucleic acids. Chromosomes have a protein core called the kinetic core, and from this protein core extends the arms of the chromosomes. I'll just draw these little lines here. So the kinetic core is here in the middle of this structure, which is called the centromere. That structure right there would be the centromere. And the kinetic core is a hook-like structure that's in the middle of the centromere. All chromosomes have a centromere. Some centromeres are in the middle, like this one, in which case that would be called a metencentric. Some are shifted off to the towards the ends, but not at the ends, in which case those are called arcocentric. And some centromeres are at the end, in which case they're called telocentric. And you don't have to know those terms. They're just used to describe different chromosomes. Also, chromosomes will have different lengths of arms. Some chromosomes will have 2 long arms, some will have 2 short arms, some will have 1 long, 1 short. And they'll have different banding patterns. If you see a chromosome that's stained with a stain called Giemsa, it'll take on a black and white banding pattern, and every chromosome has a unique banding pattern. So what this boils down to is chromosomes have physical characteristics that you can use to tell them apart. They're different sizes, they have different arms, they have different positions of the centromere, and they have different banding patterns. So that can distinguish one chromosome from another. Now we already said that we have 46 chromosomes, and that's our diploid number. Well, this is 2 sets of 23, and so the sets occur in pairs. So we'll draw another one over here, roughly the same size and shape. Okay, so those are our 2 chromosomes, and the name for these are called homologs to one another. Okay, homologs, this word here, homologs. And if they're lined up like that, that would be a homologous pair. So to make this make sense, let's say this is chromosome number 1, right? And I'll just put an M here. And here's chromosome number 1 and put an F next to that. What do you suppose the M and the F stand for? All right. I can think of one thing. What do you suppose M and F stand for in this context? Right? M stands for male and F stands for female. These are the chromosomes you got from your two parents. And when you get one chromosome from each set, from each parent, when they come together in the new zygote to make you, we call that pair a homologous pair. Now the thing about homologous chromosomes are they're structurally similar. So this means that they have the same size, they have the same position of the centromere, they have the same banding pattern, and they contain the same types of genes. And that's important to understand, that word type. This means that if this chromosome contained the genes for, let's say, eye color, which there are 7 different genes, hemoglobin, and I don't know, maybe the open reading frame for the gene to make angiotensin converting enzyme. Actually, I don't know what chromosome that's on. But let's say we had those, right? That means that each homolog would have those same types of genes. So if this gene if in this position here called a locus, we had a gene for eye color, over here we'd have a gene for eye color. We wouldn't necessarily have to have the same form of that gene, which is called an allele. Different forms of genes are called alleles. So at a locus, L-O-C-U-S, no, it's not. At a given locus, we have a particular type or category of gene. The actual form of the gene we have is called an allele. So a locus is a physical spot along a chromosome where a gene type occurs. The particular gene that is there is actually called an allele. So what I'm saying is that homologs have the same loci, but may have different alleles. Okay? Homologs have the same loci, that's plural for locus, the same loci, but they may have different alleles at those loci. I say may have because it's possible that they have the same form of the gene, in which case we would call that situation homozygous, or they could have different forms of the gene, and that would be different alleles, and we call that condition heterozygous. But I get ahead of myself. We'll talk about that in genetics in the last week of the class. Okay. So we've got these homologs, one from mom, one from dad. Chromosomes of similar size, shape, appearance, and genetic makeup, although not exactly identical to one another. That's an important concept because in meiosis, what we're going to do that is very different from mitosis is we are going to separate the homologs from one another and send only one of these pairs, or one of these chromosomes, into a gamete, not both. In mitotic division, we will take copies of both of these into the new daughter cell. So the new daughter cell will have the same genetic makeup as the parent. But in meiosis, we only take one, and that means that the gametes that are produced, the daughter cells that are produced, have a different genetic makeup than the parent. It's made out of the genetic material that was in the parent, but it's not identical. And so that's what it's getting at here when it says, no two gametes will ever be exactly alike. All right, so here's a cartoon that kind of puts it into play. These little X's here are supposed to represent chromosomes. When we talk about cell division, we talk about the different phases of them. And the phases that we have are, just listing off the names, prophase, metaphase, anaphase, which I don't see listed here, and telophase. So I'm confused by, they just kind of skip over some of those. All right. Well, so let's talk about what happens. Before I get to prophase, I guess I have to tell you something else. If we were to imagine a circle here, and it's not the best circle in the world, but let's make that a circle, and let's put a couple marks on it. Put one there and one here, and we'll put an M next to this, and an S next to this. This would be an example of something called the cell cycle. And so it's a cycle going around that direction. Okay. Whoops, not the best arrow in the world. So it's a cycle going this way. We would split it into two growth phases. This is called G1, and up here is called G2. Okay. Over here, we can have an imaginary area, which is called G0. I'll tell you why I said that was an imaginary area. So this is our typical cell cycle. We have a G1, growth period 1, separated by S, which is synthesis phase. Then we go into a second growth period, and then finally we go into M, and M stands for mitosis. The cell cycle doesn't quite apply to meiosis, and I'll tell you why in a minute, but this would be cell division, mitosis. All right, the reason I'm mentioning this now is because everything that's listed over here, mitosis and meiosis, that is solely what happens right there. Everything else that the cell does is not in the dividing period, and all of this material, G1, G0, S, and G2, is all in a phase that we call interphase. Okay. Interphase is between mitotic divisions. That's why it's called interphase. Your normal body cells spend their whole life in interphase. They don't divide. Most cells in the body are not rapidly dividing, with some exceptions, right? Smooth muscle, epithelial cells, red blood cells, well, or hemopoietic stem cells. Those are rapidly dividing tissues. Liver cells are rapidly dividing tissues. But most of our adult cells, nervous tissue, most connective tissues, bone, all of that, glandular material, most of that is not rapidly dividing. So most of that is what we call terminally differentiated. It's an adult tissue that has actually exited the cell cycle and is just hanging out here in G0. G0 is what we call the ground state, or terminally differentiated state. And that sounds terrible when you say terminal, because it sounds like it's dead. But in this case, it just means that's the end. For this cell, it's not going to go on and divide again. Most of your body cells are in G0. When you're growing, your cells, for the most part, your hypertrophic cells, the ones that are growing, stay in the cell cycle. So how this goes, when the cell is formed, it has to get bigger. It has to add cytoplasm, it has to add organelles, it has to bulk up on energy, and that's all happening during G1. That's your growth phase. When the cell gets ready to divide, the very first thing it has to do is copy its DNA. So once the cell has gotten big enough to do this, it'll have enough energy. There are enzymes inside the cell that actually are sensing the level of ATP and cellular components. And once those enzymes signal to the cell that, hey, you're big enough and you have enough energy, we can go ahead and divide, we enter into S phase. And S phase is synthesis, a DNA synthesis phase. So this is where the copying of the DNA is taking place. In order to copy the DNA, the chromosomes have to be very spread out. They have to be not so tightly packed as they would be during cell division. When the chromosomes are spread out like that, we give that a name. We call that chromatin, or it's spelled chromatin. Chromatin. Those are dispersed, spread out chromosomes. And that's the form in which they're copied in. All right, so we copy the DNA. We take one chromosome, we kind of split the strands in half, copy each one, and we make two daughter chromosomes that have exactly the same genetic makeup as the parent, barring mutation, which is something we talked about earlier in the course. So that all happens during S. When S is done, now we have two sets of chromosomes. And at that point, the chromosomes are still attached at their shared centromere. So this is how one chromosome might appear in interphase. When we copy it, we make a copy of each half, but they remain attached here at the centromere. And so it winds up looking like that. Each half of a replicated chromosome is called a chromatid. So I'll say that 2 more times. All right. Each half of a replicated chromosome is called a chromatid. And again, not the best at the mouse writing here, but chromatid. I suppose I could just insert a text box. It's going to be easier. All right. We'll put chromatids. So that's one chromatid. And there's the other chromatid. Now these chromatids are called sister chromatids. So your definition is a sister chromatid is one half of a replicated chromosome. Now, are sister chromatids identical to one another? What do you think? All right, hopefully your answer is yes, since one is copied from the other. They should be genetically identical to one another. Again, barring the fairly rare instance of a mutation, which we're going to ignore for right now. So we'll say that they're genetically identical. So those are created during S phase. And if you look over here in this prophase cell, that is the condition that chromosomes are in. See how they look like tiny little X's? Well, if we come up here to the top and we look at this cell, this would be an interphase cell that they're calling the mother cell. It says before chromosome replication. So this would be in G1. Okay, and you'll notice that those chromosomes are not little X's. They're just little bars with a centromere in the middle. All right, so we go through S phase, we copy that genetic information, and now we look like this. We look like little X's. But we're still spread out. We're still in chromatin form. So now we go through G2. G2 is the second growth period, but this growth period is dedicated to bulking up to be able to withstand cell division. Kind of think of it like this. Think you're going to pack up your house and move to the other side of the planet. Okay. Well, there's going to be some travel involved in there, and while you're traveling, you don't have access to all the goods and services that your house would ordinarily provide. So you're going to have to make sure you have enough money, you have enough food, you have enough clothing, everything readily available so that you can make that move. In a similar way, G2 is a period where the cell is bulking up and storing energy and nutrients and amino acids and nucleotides and everything it needs to accomplish cell division. Because once it starts down that path, it doesn't have time to stop and make more membrane and make more proteins and garner more energy. It has to complete division. And if it fails to complete division, it's kind of like you failing to get across the Atlantic Ocean. You're probably not going to survive. And that's true. If the cell starts to go through division, starts to go through mitosis or meiosis, and runs out of energy or runs out of organelles, it'll die. So that's the importance of G2. Now we have what's called a checkpoint, a G2 to M checkpoint, where the cell actually makes sure that it has enough energy to accomplish division. If it doesn't have enough energy in cellular components to accomplish division, it's halted, and it won't be allowed to go on and do division until it does get enough material, and then it'll go ahead and divide. So that's the G2 to M checkpoint. There's also an S to G2, well, it's really G1 to S checkpoint, where we make sure that we have enough energy to copy all of the DNA. So we have these two different checkpoints. If the checkpoints are not satisfied, like I said, then actually programmed cell death is stimulated. And there's a fancy word for that, of course. It's called apoptosis. Apoptosis is programmed cell death. And if a cell doesn't have the necessary components or energy, or there's some damage to the DNA, there are genes called tumor suppressor genes that are in charge of activating the apoptotic pathway. And if we don't satisfy those genes, those checkpoint genes, via something called cyclins, then they stimulate apoptosis and the cell will die. It's better for the cell to die than to mess up during division, because that's going to cause disease if that occurs. Okay. So the bulk of this time is spent in interphase, and that's what we're showing here. This is an interphase cell. We've replicated the DNA, so replication of DNA also happens in interphase. When we satisfy the G2 to M checkpoint, then we start mitosis, or meiosis. The first phase of mitosis or meiosis, and I'm just going to say the first phase of cell division to keep that simple, is called prophase. And a lot of interesting things happen in prophase. Prophase is the most action-packed of the phases of meiosis. And in fact, you can break it down into 5 or 6 different phases just of prophase. And we're not going to do that, because it's not necessary for us to go into that much detail. But I would like you to make a little list of what happens in prophase. The first thing that happens in prophase is the chromosomes condense. They compact. They go through 3 different levels of DNA compaction. And again, you want to take a genetics class, I'll tell you all about that, but right now we'll just keep it simple. The chromatin, which is the spread out DNA, gets twisted and coiled and packed into a really tightly compacted chromosome. It's more than 300-fold compaction that you have occurring. So that takes a single chromosome, which if it were stretched out in a linear fashion, would be nearly a meter long, some of these. Okay. We take that and we compact it down to something that's really only a micrometer or so long. Maybe 3 or 4 micrometers at most. So that's called compaction, and that's what we see here when those chromosomes look like little fat X's. That's the compacted DNA. All right, so that happens at the beginning of prophase. Well, what else happens in prophase? Well, there's an organelle inside the cell that perhaps we remember is called a centrosome. And really, up to this point, we've never talked about much of a role for the centrosome. Within the centrosome is a tiny little proteinaceous body, and here you can kind of see it in yellow down there, called the centriole. And the centriole is an example of something we call an MTOC, and you don't have to know that term, but it just stands for microtubule organizing center. The centrioles are the nucleus, and I don't mean nucleus as in the cell nucleus, I mean a nucleus as in something that gives rise to something else. It's the starting material for growing a microtubule framework. So the centriole grows what's called the mitotic spindle, or the spindle apparatus. It's a microtubule network that extends from end to end of the cell. So what we have to do in prophase is the centrosome splits into two. So we get two centrioles, and those centrioles move to the opposite sides of the cell, establishing what we call the poles, the two ends of the cell. And then the microtubules grow between it and make a spindle apparatus. So you've got one end here, one end there, and you've got the mitotic spindle that grows in between, microtubule network. So that all happens in prophase. All right, this nice nucleus that we've had with a nuclear membrane housing the chromosomes and protecting it from all the harsh cytoplasm disappears. The nucleus actually breaks down. And I know it's going to sound like a made-up word to you, but the nucleus sort of bubbles and boils and breaks, and that's a process called blebbing. The nucleus just sort of disintegrates. Now that doesn't mean the material in the nucleus is destroyed. It just means the nuclear envelope breaks down. And it has to do that because we're going to have to separate these chromosomes. So in order to do that, we've got to have the microtubules have access to them. All right, so we have the nucleus breaking down, the chromosomes are condensed. And then something very interesting happens in meiosis. So everything we've talked about is shared between mitosis and meiosis, but something unique happens in meiosis during prophase that does not happen in mitosis. As you can see here in meiosis, the two X's of similar size and shape line up next to each other. They pair, in other words. And that is called homologous pairing. That does not happen in mitosis. Okay. In meiosis, during prophase, the homologs, in other words, chromosome 1 from mom and chromosome 1 from dad, pair. Now, if we think about this, the chromosomes are already replicated, and so each chromosome consists of two sister chromatids. So you've got one homolog and the other homolog, and then they come pair each other, and they kind of wrap around one another in this funky little process. That's called tetrad formation, and the process of them touching like that is called synapsis. So tetrad formation is the two homologs coming together, and they intertwine around each other, and that's called synapsis. Something kind of fascinating happens during synapsis. The chromosome pieces of the sister chromatids, of the non-sister chromatids, so this piece from this homolog and this piece from this homolog, can actually break off and exchange. It's called reciprocal recombination or reciprocal exchange. And I'll show you a diagram of that in class when we talk about genetics. What that does, though, it's a very powerful phenomenon, because remember, you got each homolog, one from mom, one from dad. When they come together like that, and let's just say I exchange the tips of my fingers here, I would exchange those pieces and then they would separate again. What was on this hand is now over here, attached to this chromosome. That's called recombination. Remember, the homologous chromosomes have similar loci, but they have different alleles at those loci, or at least they may have, which means when they swap pieces in this reciprocal exchange, I get a shuffling of genes now connected to each centromere that I never had before. And at the end of this process, I'm going to separate those homologs from one another into separate cells, and that'll give each homolog a different shuffling of genes. Okay, it's called genetic recombination. So the first step in this is this tetrad formation with synapsis that occurs during prophase, where the homologous chromosomes pair, and then during a process called crossing over, that's what that reciprocal exchange is called, we can have bits of the non-sister chromatids, in other words the homologs, break off and exchange. And you can have along the length of a chromosome, maybe as many as half a dozen crossover events occurring. Okay, so we have crossing over occur in prophase. And then we move on to the next phase. The next phase of meiosis is called metaphase. And what happens in metaphase is the chromosomes line up at the equator of the cell. And they're pulled there by attaching to the microtubules of the spindle apparatus. Now here's another important point about metaphase. When the chromosomes line up at the metaphase plate, or the equator of the cell, they do so such that one homologous pair is on each side of the metaphase plate. So if we imagine the metaphase plate being a dotted line right there in front of me, here would be one homolog, and there would be the other homolog, and they're on either side. And they could be separated. That's what's going to happen in the next phase. If you take a glance over at mitosis, you take a look at that, you'll see that there's no homologous pairing there. The chromosomes line up at the equator in mitosis, but they do so such that the sister chromatids of a replicated chromosome are on either side, and they're going to separate in the next phase. So that's a big, big difference between mitosis and meiosis. So in meiosis, which is what is happening in the gametes, in metaphase 1, which is the first round of meiosis, remember meiosis is 2 rounds of division. In metaphase 1, the homologous pairs line up at the equator. Go on to the next phase. The next phase is called anaphase, and here they don't really show it. They don't bother showing. They just say here homologous chromosomes separate, but sister chromatids remain together. We pull these chromosomes apart, and that's called anaphase. This chromosome goes that way, and the green chromosome goes to the left, blue goes to the right for the upper pair, and just the opposite of that for the lower pair. Right. All right, sorry for the little interruption there, but I'm back. So I think I was saying that the homologous pairs, they separate from one another in a phase called anaphase, and that's really all that anaphase is. You just see the chromosomes moving to the poles. Then we get to something called telophase, and telophase is the end of meiosis 1. Telophase is kind of an upsetting phase to learn about because it's this weird hybrid phase where you may or may not fully go into it. So let me explain what happens. Essentially, when we get to the end of division, the chromosomes have moved to the poles. At that point, if the cell is going to take a little breath before it goes through the second round of division, called meiosis 2, then we have time to let the chromosomes spread back out into chromatin, so we call that decondensing. The nucleus can reform. But if we're going to roll right into the second round of division immediately, then none of that happens. The chromosomes don't decondense, and the nucleus doesn't reform. But what will always happen in telophase is the cytoplasm will divide completely. So we'll get 2 plasma membranes separating these 2 daughter cells. Now, in reality, the 2 daughter cells are snugged up right next to each other, but they've formed a new plasma membrane between them. So at this point, at the end of telophase, we have 2 distinct cells. So we have daughter cells formed. And the most important thing is if you count the chromosomes, you'll see that the daughter cells formed in telophase have 1, 2 chromosomes. If you come back up and look, well, in this picture they have 2 chromosomes. If you come back up and look at the parent cell that we began with, there were 1, 2, 3, 4 chromosomes. I always tell students when you're counting chromosomes, count the centromeres. Don't worry about the chromatids or anything else, just keep it simple. Count the centromeres. If you have 4 centromeres, you have 4 chromosomes. So even here when they're X'd like that because they're replicated, the sister chromatids are still joined at a common centromere. So there's still 1, 2, 3, 4 4 different chromosomes. And you come down here and you've got 1, 2 different centromeres. So there you have 2 chromosomes. So that's it. We've reduced the chromosome number from 4 down to 2. Now this is a drawing, so keep in mind that in human cells we wouldn't have 4 chromosomes, but we'd have 46. And after meiosis 1, we'd be down to 23. So all we've got to do now is go through meiosis 2. And in meiosis 2, what we do is, well, we don't do any replication, because we did that in interphase. So now we take those same chromosomes, and we put them back into prophase again. So we go into what's called prophase 2. And in prophase 2, if the nucleus reformed, it breaks down. If the chromosomes decondensed in telophase, now they recondense. The centromeres, the centrioles are going to have to separate and move to the poles. A new mitotic spindle is going to form, or spindle apparatus is going to form. So that all happens in prophase 2. There is, however, no tetrad formation, no synapses, because we don't have homologs anymore. The homologs have been separated. So that doesn't happen. Then we roll into metaphase 2, and in metaphase 2, the chromosomes line up at the poles with each sister chromatid on each side. And then in anaphase 2, the centromeres unhook, and we actually separate the two sister chromatids from one another. So now we have chromatid 1 going to one cell and chromatid 2 going to the other cell. Now, if we had not had crossing over, those two chromatids, the two halves of a replicated chromosome, would be identical to each other. And when they move to the poles, we would just get the same information in both cells. But because crossing over occurred, and those chromatids exchanged pieces with their homologs, now they're no longer genetically identical to one another. They have bits and pieces of the other homologs stuck in there, and it's kind of random which ones went where. So now we have two genetically distinct sister chromatids, and we separate those. What we've achieved is each chromatid has the same loci, they have the same types of genes, but they may have different alleles as a result of recombination that happened in prophase 1. So now we separate it, and we wind up getting 1, 2, 3, 4 gametes produced, each one containing chromatids that have a slightly different makeup than any of the chromatids we had up here in the parent cell. And these gametes that are produced, well, these are the spermatids or the ova. And when we get into female reproduction, we'll talk about a difference here. In males, in spermatogenesis, we make 1, 2, 3, 4 spermatids. In females, in oogenesis, the production of an egg, 3 of these are degenerate. They break down. One of them is a very large egg cell. So while in spermatogenesis we make 4 gametes, in oogenesis we only make 1 gamete and 3 what are called polar bodies. All right, the next several slides just kind of talk about the terminology that I already mentioned. We know that in prophase 1, one of the main events that has to happen there is a synapsis idea where the homologs come in pair. That's called a tetrad because there are 4 chromatids involved. We can have points of contact where the non-sister chromatids touch one another for the reciprocal exchange. That touching can show up in a light microscope, and we can see that as a chiasmata, a cross. Right? And that's where crossing over can occur. They call them between male and female chromatid. Well, that's what I mentioned as one from your mom and one from your dad. The chromosomes themselves do not have a sex. Okay, you don't have a male and a female chromosome. It's just one from mom, one from dad is what that means. So I already mentioned these things. All right. Oh, here's finally I get to the right slide. This is the slide that shows all the phases. So there's prophase 1, and I'm not going to recap everything I said that happens. You can read over this. Prophase 1, metaphase 1, anaphase 1 with the separation of the chromosomes. I will mention, though, the separation of the chromosomes, that I'm not sure if I gave that term. It's called karyokinesis, the division of the nucleus, karyokinesis. That happens in anaphase. In telophase, we get cytokinesis, the division of the cytoplasm. And in truth, cytokinesis begins in anaphase, but it ends in telophase. Karyokinesis, the division of the chromosomes, begins and ends in anaphase. Then we go to prophase, meiosis 2, which begins with prophase 2, metaphase 2, anaphase 2, telophase 2. And that's where we generate our gametes. Okay. All right. So now I want to step outside of the lecture slides a little bit and show you the lab slide that we have that goes with this. This is our meiosis board that we have from lab. Let me pick up the pen here. What I expect you to know about what we see here is that this cell that starts right up here, this would be our best representation of an interphase cell. Remember, that's the part of the cell cycle that the cell stays in most of the time. It's the growth, then the synthesis phase, then another growth phase. It also includes a terminally differentiated G0 phase. Okay, so how do we know it's an interphase cell? Well, look at the chromosomes. What do you see about the chromosomes there that tells me that that must be an interphase cell? In fact, it must be a G1 cell. Well, if you said the chromosomes are not replicated, you are right. Okay, the chromosomes are not currently replicated. They're just single arms coming off of that central little centromere. When we go over to this slide, now you can see the chromosomes have this X-like appearance. So they've clearly been replicated as they went from here to here. So the middle box at the top represents a prophase 1 cell. So interphase here, prophase 1 there. Now there are a few things that I have to pick with this model. One is that it shows the chromosomes condensed, and they would not be condensed like this before prophase. They'd be much, much longer than that. But that's a minor issue. Also, here in prophase, they're showing an intact nuclear membrane. And so that wouldn't be quite accurate either. This dark area is supposed to be the nucleus. That nuclear membrane would break down in prophase. So in prophase, let's go over the list of things we would expect to have happened here. The centrioles would have moved to the poles, and actually you can see that there and there. Okay, there are the centrioles. And what do you suppose these blue lines are going from pole to pole? If you said the spindle apparatus, you would be correct. Okay, that's that spindle apparatus made out of microtubules. Well, that actually forms in prophase here. What is happening here? What's the significance of that pairing I just put the box around? The green with the yellow. Do you understand what the green and the yellow mean? The green would indicate, say, the maternal chromosomes, the one you got from your mom, and the yellow are trying to indicate the paternal chromosomes, the one you got from your father. Right? So what is that condition called? The green and the yellow that have similar size and shape and banding pattern and chromosome and centromere position. What is the name for that? If you said homologs, you would be right. That's a homologous pair there. So we have two homologous pairs. One a little smaller and the other a little bigger. So right now we have synapsis occurring, tetrad formation with synapsis. Now what it's not showing you here is that the chiasmata would be forming where the non-sister chromatids would be connecting, and then they could be crossing over, they could be exchanging pieces, and we get a reciprocal exchange. And it's a reciprocal exchange. That one piece breaks off and sticks over here, but one, say from my knuckle up would break off here, and then from the knuckle up breaks off here, and then they exchange those pieces. And when they separate, now I'd have, this part of this finger over here with that part, and vice versa. So that's a reciprocal exchange. So once we've achieved the crossing over and the other functions that happen in prophase, then we're ready to go to metaphase 1. So that's what we are in here. This is metaphase 1. And so right here would represent the metaphase plate, and that's called the equator of the cell. And we can see that the homologs are lined up on either side of the metaphase plate. And that's really what happens in metaphase, the lining up of the, in meiosis, the lining up of the homologous chromosomes at the plate. And that's to prepare the way for anaphase. So here's anaphase down here, and the chromosomes are now moving in opposite directions. And they're moving because they're being pulled, or I guess actually physically it's pushing. This grows here and it pushes them apart. So let's just think of it conceptually, that they're being pulled to the poles. And that's the separation of the nucleus, so that's called karyokinesis. These chromosomes moving to the poles is called karyokinesis. You can see the beginning of the formation of a cleavage furrow. An actin band forms around the cell like a belt, and it starts to tighten up and cut the cell in half, and that's called a cleavage furrow. That's the beginning of cytokinesis, the division of the cytoplasm, which we know that has to complete by the time we get to the next phase, which is telophase. So here, this is a situation where we could call this telophase 1. It's a T. We could call that telophase 1, and then we could put slash prophase 2. All right. So we could call it telophase 1 or prophase 2. I don't know if that notation helped at all because it's so messy. And the reason I say it could be either or is because, like I said, the events are a little indistinct. What we know has to happen in telophase is that we have to get division of the cytoplasm. In other words, cytokinesis has to complete to give us our two daughter cells. Here's one over here, and there's the other over there. Count the chromosomes, guys. And how do we count the chromosomes? Remember, that's counting the centromeres. One here, one there. So there are two chromosomes here. Back up here in metaphase, we had 1, 2, 3, 4 chromosomes. So clearly going from metaphase to telophase, we went from 4 chromosomes to 2 chromosomes, and we did so precisely by taking 2 sets and turning that into 1 full set. So we went from diploid here to haploid there. So telophase 1 / prophase 2, the cell is haploid now. And that's why we call meiosis 1, the first round, the reduction division. Okay, we've reduced the chromosome number from, in humans, 46 to 23. Pardon me. From here on out, it's pretty straightforward. Meiosis 2 works exactly the same way as mitosis. We take the chromosomes and we line them up at the metaphase plate such that now the sister chromatids, and I'm not doing a real good job drawing that. Sorry, this one's a little straighter. Okay. Such that the sister chromatids are on either side of the metaphase plate, and now they're going to separate. And they're going to go their happy ways to the opposite poles. But that doesn't happen yet. They just line up at the equator. So the sister chromatids line up at the equator during meiosis 2, metaphase 2. In anaphase 2, they separate. And finally, telophase 2, we have cytokinesis, and we've divided those cells into 4 gametes down below. So things that I like to ask on here, let's go over our list to make sure I've shown you everything. We saw the centrioles. Okay, and then they're shown there and there. We know this is an interphase cell. Oh, so in spermatogenesis, the cell that's going to divide to give you spermatids is a spermatogonia. That's your interphase cell. So this would be a spermatogonia. Once we start dividing by meiosis, the spermatogonia are called primary spermatocytes. So once they start dividing by meiosis. Before they divide, they're called spermatogonia. Once they start dividing by meiosis, they're called primary spermatocytes. Spermatogonia can do 2 things. One, they can divide by meiosis to give you spermatids. The other, they can divide by, well, I want to make sure I said that right. They can divide by meiosis to give you gametes, and that's what this board shows. Or they can divide by mitosis, somatic cell division, and if they divide by mitosis, they just regenerate more spermatogonia. Okay? And that's a big difference between what males and females do. Males are doing ongoing spermatogenesis constantly. They produce millions of sperm a day. Females, on the other hand, they're born with about 400,000 oogonia, the cells that are going to give rise to eggs. Through atresia, by the time they get to puberty and degeneration of the oogonia, they only have about 40,000 left, and they never regenerate anymore. So, and that's fine, because during the entire reproductive age of a female, where maybe she's going to go from early to mid-teens to late 40s, early 50s, you're only going to ovulate 1 egg a month. So 40,000 is a plentiful supply for that entire span. Whereas males producing millions a day, they would very quickly run out of spermatogonia, so they have to replenish their spermatogonia constantly. Okay, let's see here. And I think we've covered everything else in that list. Oh, not quite. So once we go through meiosis 1, we get down here and we'd have a prophase 2 cell. Remember, that's the end of the first division. So the cell at the beginning of the second round of meiosis is called a secondary spermatocyte. So before we divide by meiosis 1, it's called a primary spermatocyte. And before we divide by meiosis 2, we're called a secondary spermatocyte. And finally, the gametes down here in males would be called spermatids. So I just have 2 blow-ups showing you meiosis 1 and meiosis 2. Here's meiosis 1, and again, it's called the reduction division because we are going from 2N to N, all right? Diploid to haploid. And we do that by dividing the homologous pairs, okay? So in meiosis 1, the homologs separate. That's the money line you want to remember. Meiosis 1, the homologs separate. Now I mentioned something here, the law of segregation and the law of independent assortment. You may or may not have heard of these before. An Austrian monk back in the mid-19th century came up with these laws. His name was Gregor Mendel, and he came up with 7 major laws of inheritance, and these were 2 of the most important ones. Let's do the law of independent assortment first. All this says is that where one homolog lines up, or on which side of the equator one homolog lines up, has no influence on the second pair. Meaning that if the yellow one is on the right, that there's a 50-50 chance that the yellow one of the next homologous pair could also be on the right. Now in this diagram, they've put it on the left. indicating again that the pairs don't have to stay together. So, or not the pairs, the sets. The sets don't have to stay together. Remember, the yellow set is the one we got from, what did I say, I think Dad. Okay, if this was the paternal set, we don't have to inherit those together. That's what independent assortment says. It says that one homologous pair does not influence how another homologous pair lines up at the equator in metaphase 1. Right. The law of segregation tells us that the alleles that are on here, so there's an allele here and there's an allele here. The alleles separate from one another. Okay, they separate from one another, and that doesn't happen. Well, actually, it does. It happens right here. Okay, as a result of them separating, that's what segregation means, to separate. So Mendel figured out that while genes do occur in pairs, that was another one of the things he figured out. Genes occur in pairs, and that's because we have two homologs. He figured out that they separate from one another during sex cell division, so these pairs are not inherited together, and that's called the law of segregation. All right, meiosis 2. So again, meiosis 2 just mimics the steps of mitosis, and so we start with a prophase cell, condensed chromosomes. The chromosomes line up at the equator such that the sister chromatids will be pulled apart. In anaphase, the sister chromatids come apart at the centromere. So again, if you follow the concept of counting centromeres, here we have 2, 1, 2, so we have 2 chromosomes. 1, 2, 2 there. Now here they're separated, so I guess temporarily you could say that this one now has 4 chromosomes. But we're doing cytokinesis, we're dividing that cell. So by the time we get to telophase, we're back to 1, 2. So what does meiosis do for us? It takes us from having 2 chromosomes to making 2 daughter cells, each with 2 chromosomes. The difference is up here we have 2 replicated chromosomes, and down here we have 2 single chromatids in each cell. All right. We'll continue on a little bit here. I think I'll try to finish up this section. Once meiosis generates the spermatids, that's fine. That's called well, gosh, what's that called? Yeah, that's called spermatogenesis. That's the generation of the spermatids, which involves meiosis. Okay. Now what we need to do is we need to take that spermatid that we formed and turn it into a sperm cell, and that's called spermiogenesis. Threw me for a loop here. I was looking at this term. I didn't recognize it immediately. A spermatogenic cell, well, the term I gave you for that was just this guy. It's a spermatogonia. So they're a type of pluripotent stem cell that can give rise to more of themselves or to become spermatocytes. And I just talked about that a second ago. Spermiogenesis is taking those spermatids that we formed at the end of meiosis and turning them into sperm. Here's a picture of a seminiferous tubule, and I'll show you some histology slides at the end of this presentation. Well, actually, I'll make a separate presentation that shows you the reproductive slides. This is where spermatogenesis takes place, and just to reiterate something we said earlier, this is the tubule, a seminiferous tubule. These cells out here are a mixture of myeloid cells, the smaller ones, and these larger ones are the spermatogonia. And the spermatogonia start dividing, and they become spermatocytes, which I imagine that yellow one would be a spermatocyte. And they go through meiosis 1, and then down here they go through meiosis 2, and they generate these little guys down in this area. There. And those are spermatids. And the spermatids gradually go through a process called spermiogenesis, where they convert from a little cell like that, with no noticeable tail, to guys like this that now have a small head, a long midpiece, and a long tail. And those are sperm cells. And then these areas that are kind of shown in like a pink, that material, that's actually a nurse cell or a Sertoli cell. And here's a diagram, a cartoon showing you what's going on there. So there's our spermatogonia. And these are the ones that can divide either by mitosis to regenerate more of themselves, or divide by meiosis as they move down here to the lumen. They call those A and B daughter cells. That's not a nomenclature I hear very commonly used, so I wouldn't worry about that. Just know that the A cells, what they're calling A cells, are just a replenishment of the spermatogonia. The B cells are the ones that are going to become spermatocytes. Remember, these cells over here are called cystinocytes or nurse cells or Sertoli cells, and they're the ones that make all of this kind of orangish material, and they're formed. They have gap junction, not gap junctions. They have desmosomes at their lateral borders that form the blood-testis barrier. So we keep the immunologically distinct spermatids away from any antibodies that could get in here and destroy them. Okay. So the process going from this compartment out here, which is called the basal compartment, to then this compartment down here, which is called the adluminal compartment, and you see those terms right there, basal and adluminal, is meiosis. So meiosis is occurring as we go down through here. So here's our primary spermatocyte that's going through meiosis 1. Once it's completed meiosis 1, then we're in to meiosis 2. And then when we complete meiosis 2, there are spermatids. And the spermatids slowly go through morphogenesis, and they go from looking like a little daughter cell like this to starting to look like a sperm cell. And I say starting to look like a sperm cell because this process here won't actually happen and finish until we get into the epididymis. Okay, so we know what a spermatogonia is. I said don't really worry about the A and B distinction. We went over meiosis 1 and meiosis 2 already. This is just showing you the reduction division concept of going from diploid to haploid. I won't dwell on that a whole lot. And we got to convert a spermatid at the end of meiosis into a sperm cell. And so this is the morphogenesis that takes place, and you really don't have to worry about anything here. We grow a cap on the end of the spermatid, which is called the acrosome, and that's going to be important for fertilizing the egg. The acrosome contains digestive enzymes and receptors that are capable of specifically interacting with the egg cell vitelline membrane. So that's called the acrosomal reaction, and that has to happen to have fertilization, so that's important. Then we have the nucleus, where of course we house the haploid genetic information. We have a mid-piece that contains tons and tons of mitochondria. So they pick up fructose from the nourishing fluid from the seminal fluid, and they bring that in and they burn it and they make ATP, and that just drives the motor. The tail here is a big flagella. Interestingly, this is the only flagellated cell known in human life cycles. Okay, it's the sperm cell. And here, this process takes up to 24 days for this to occur. And then you add that to the 40 days that we might spend in the epididymis for capacitization, and you're up to about 60 days to generate a sperm. Okay, so that just reviews the pieces of the sperm cell. The role of the Sertoli cells. So Sertoli cells, those are those big, large nurse cells. And as I mentioned, they form the blood-testis barrier, which I'm sure that's in here somewhere. I'm just not seeing it. Is there another slide? Yeah, well, that's what this is. Yeah, so we divided into two compartments. That's the blood-testis barrier. All right. As far as providing nutrients and signals to the dividing cell, this is important. Okay, the Sertoli cells are what respond to testosterone, and then they actually convert the testosterone into a form of estradiol that the developing spermatocytes need in order to finish meiosis. So that's the nutrients I was talking about here. And the signals are growth factors that are actually the result of cell-to-cell contact from the Sertoli cells to the spermatocytes. So the spermatogonia will not go through meiosis unless they're coached to do so by Sertoli cells responding to testosterone coming from the interstitial cells, which are on the outside of the tubule. The Sertoli cells can actually help push the developing spermatocytes into the lumen, and they secrete some fluid to help wash them into the epididymis. And apparently the Sertoli cells can reabsorb spermatocytes and germ cells that are faulty. The cystinocytes, i.e. the Sertoli cells, form the blood-testis barrier. And that's the same picture we saw before. Oh, there we go. There's a slide that talks about it. Perhaps we remember that any antigens that are self-antigens are selected against during negative thymic selection during fetal development. But since sperm are not produced until puberty, they're not present to have their antigens recognized as self. So that means that we will make antibodies against our sperm. So we have to keep them isolated in a compartment where the antibody-producing cells do not have access to them. So as I mentioned, you take the 20-some days that it takes to generate a spermatid, and you couple that to another 40 days for capacitization to occur in the epididymis, and you're up to about 60 days for the generation of a functional fertilization-capable sperm. Some causes of infertility. I don't hold you responsible for any of this information, but you might want to read over it. All right, we will stop right there, guys. Let's see here.